The beginning. All right, here we are. You ready?
Thanks. Oh, yeah, it's recording now.
Marvelous. Okay, well, thanks so much for everybody for coming to the cognitive neuroscience last discussion of the day of the year, as it were. We wanted to have a chance to dig into dopamine a little bit more and what its role is in the brain. I'm taking a somewhat combined approach in that I'll be talking both generically about things that hopefully you all know, but also trying to get into a few more refined ideas that you may or may not know about dopamine already. So and I am not an expert. I'm going to say right now, I don't have any bio or med or pre-med background. So everything I've learned about dopamine, I've sort of had to pick up on my own, much to the embarrassment of many of my collaborators. But I'm encouraged nonetheless, because it is a really fascinating molecule. So we wanted to talk about dopamine because, as many of us know, it's certainly been implicated in psychosis, implicated in depression, implicated in attention function and dysfunction in ADHD, and obviously a known role in neurodegenerative disorders and the motor problems that come along with Huntington's disease, Parkinson's disease, etc. So there's this wide range of effects that dopamine has in the brain, and the question is, how does it do what it does? How can dopaminergic issues lead to hallucinations in one case and the inability to move in another case? What's actually happening there? So quick overview of dopamine. Well, when I was little, it was considered that dopamine was really the pleasure molecule. And you'll still see that if you sort of look on YouTube for introductions to dopamine, they'll talk about its role in the sensation of pleasure. But it's really involved in sort of motivational salience and the bringing signals to mind, either positive or aversive, paying attention to those and making sure that you focus on those stimuli. So the dopamine is really 80% of the catecholamine content in the brain is dopamine, but dopaminergic neurons are really not particularly common. And I sort of overlaid this diagram here to show you where the dopaminergic neurons are really concentrated largely in the prefrontal areas, nucleus accumbens, hippocampus, the striatum, and of course the midbrain. And really there's a relative paucity of dopaminergic neurons elsewhere in the brain. So they are very concentrated in specific regions, but they communicate all over the brain. So the dopaminergic effects and what happens in these dopaminergic neurons has effects all over every neural system, really. So there are several dopaminergic pathways. Hopefully everybody remembers that from introductory classes. The ventral tegmental area, of course, is where the dopamine is largely produced. There is a mesolimbic pathway, which has been implicated in pleasure and reward-seeking behaviors, addiction, emotion, etc. There's a nigrostriatal pathway heading into the striatum, involved in movement and sensory stimuli. And then there's the mesocortical, which also starts in the ventral tegmental area and projects to the rest of the cortex. And it was involved, of course, with everything that the cortex is doing. And there's another one that involves the nucleus accumbens, which was highlighted, I think, in one of the papers. That goes, yeah, obviously goes through the hypothalamus. And so there's a number of dopaminergic pathways, and they do very different things, and they project to very different areas, which accounts for some of the breadth of what dopamine can do. And an important point to keep in mind is the different dopamine receptors and what they do and how they behave. Now, there's up to five or more of these dopaminergic receptors, but they really fall into two families, two primary families: the D1 family and the D2 family. So this is a D1 dopaminergic receptor on an excitatory pyramidal neuron, a D2 receptor on the inhibitory interneuron from the mesocortical pathway. This is a sort of a cartoon of a D1 dopaminergic receptor on the medium spiny neurons in the striatum, and of course the D2. And so these different receptors can play different roles if they're on different neurons. And so the D1 receptors tend to be on these excitatory pyramidal neurons. The D2 receptors tend to be on the inhibitory interneurons. The medium spiny neurons tend to be both D1 and D2, but there's a lot of D1, slightly less. And so the different balance of what these receptors do really can play a huge role in understanding the dynamics of these circuits and the role of what these circuits can actually do, and the downstream effects of what they can do. So just a quick overview again, some of the differences between the mesolimbic dopaminergic pathway from the ventral tegmentum through the nucleus accumbens and the mesocortical dopaminergic pathway. The mesolimbic pathway, the projection is quite robust in its density, whereas the cortical outlay is really more sparse. The location in the ventral tegmentum, it comes from different areas, the lateral versus the medial. The firing rates and the dynamics of the firing rates are very different across these two pathways. The dynamics of the dopaminergic uptake or breakdown in the synaptic cleft is very different. Are there autoreceptors on the neurons that would pick up the dopamine? Yes in one, no in the other. What are the responses to reward or punishment? You can see those are very different between the mesolimbic and the mesocortical. And of course, the inputs to those networks are very different as well. So even though we look at this and go, okay, well, the main difference is where it projects. No, there's actually quite a lot of other differences as well between the dynamics of these networks, their responsiveness to positive or negative stimuli, etc. That can really, and you'll see that later when that some of these differences really play a role in how we think about these neurons as well. So a basic finding regarding dopamine and psychosis, which of course is what's near and dear to my heart, is the increased dopamine or hyperdopaminergia in the striatum, right? So if we come back to here, right, we're looking at in the striatum. So in this nigrostriatal and mesolimbic areas, we really have hyperdopaminergia. But at the same time, we have reduced or hypodopaminergia in the frontal cortex. So in our prefrontal areas where we have most of our dopaminergic activity, we're actually reduced in schizophrenia as opposed to the increase in the striatum. And of course, reduced connectivity among these regions between the striatum and the frontal cortex, between the basal ganglia and the frontal cortex, the subcortex and the cortex. Not particularly specific to these regions because within schizophrenia, part of what we see is sort of a reduced connectivity in many cases. Overall, it's not uncommon to see reduced connectivity. But one of the main points of the paper that I wanted to make sure you paid attention to was these distinctions among different frontostriatal loops. So we say, oh, frontal striatal loops. Well, there's several of them happening here, and it's worth keeping track of what the dopaminergic pathways are doing in each of these cases. So we have at the sort of level of the thalamus, we have different nuclei within the thalamus. So we have the ventral anterior and medial dorsal thalamus and the ventrolateral thalamus that are doing different things. Within this region or this loop, we're really looking at the dorsolateral prefrontal cortex, superior frontal regions up here, and the dorsal caudate within the striatum, and then the ventral anterior and medial dorsal thalamus, and this all, which then loops back to the DLPFC. So that's one particular loop that's really involved with sort of associative, more cortical thinking. More cognition. We also have the anterior cingulate and the medial prefrontal areas. We all know how these play a role in the limbic system and the ventral striatum, also the pallidum and the striatum and the reticular part of the substantia nigra, and the ventral anterior. So these two regions are sort of overlapping in these two circuits, but then the distinction is really the dorsal versus the ventral striatum, and then where in the frontal cortex it actually goes. And then we have a motor circuit with the ventrolateral thalamus, the striatum, and the substantia nigra, the dorsal putamen, and the supplementary motor area in the motor cortex forming a loop. So all of them are involved in these striatal and cortical areas, but they're doing very different things and playing very different roles. And a large part of what the paper was talking about is how each of these might play a role in psychosis. And I think that's probably pretty clear to everybody as it is. One of the highlights, of course, is showing that this dorsal caudate within this associative loop, so that red loop that we were talking about on the previous slide, starting from the dorsal caudate, there's really a confluence of studies, so not just a single study, but a confluence of studies arguing that there is a reduction in connectivity between the dorsal caudate and the frontal cortex, not only in first episode psychosis, but also in first degree relatives who are not affected by psychosis, but that carry the genetic risk for psychosis, and also for individuals who are at an at-risk mental state or possibly at high risk of developing psychosis. We see a similar reduction. And then in individuals who have psychosis, there's also a relationship with increased positive symptoms, so that the connectivity between this dorsal striatum and the frontal cortex is reduced, you see increases in the severity of the disconnect with reality, right? The hallucinations, the delusions, etc., can be related to that level of disconnection as well. Now, of course, how does this actually happen? In the papers that we were talking about or reviewing for today, they've talked about the dual loop hypothesis. So one loop is really focusing on the role of the midbrain through the striatum. The other loop is really the mesocortical loop, going more into the cortex, and trying to explain both the striatal hyperdopaminergia and the frontal hypodopaminergia. And they focus on this idea of the accelerator and the brake, so that the glutamatergic inputs are, of course, accelerating the dopaminergic activity, leading to the hyperdopaminergia within the striatum with its downstream effects in the cortex. And then the brake, which is, of course, the glutamatergic inputs into the midbrain, are actually activating the GABA, which then is inhibiting the dopamine in the midbrain, which goes to the cortex, as opposed to the dopamine in the midbrain that goes to the striatum. And of course, knowing that the ventral tegmentum areas that project to that lead the dopaminergic projections, both striatally and cortically, knowing that there are different areas in the ventral tegmentum, you can see how we can have two separate effects within the ventral tegmentum that actually lead to increases in one dopaminergic pathway and decreases in the other. A different way of looking at it, one of the cartoons that I rather enjoy, this is from a Frontiers paper review from a few years ago. They're really pointing out that within the mesolimbic pathway, so from the ventral tegmentum into the limbic areas, these are the areas that really lead to the overactivity, the higher, the increasing hyperdopaminergia is really related to positive symptoms in schizophrenia, generally speaking. And of course, this would also include that ventral caudate that we were talking about. But then there's also the idea that the reduction in frontal cortex dopamine, or the dopaminergia in the mesocortical regions, are really related to negative symptoms, the mood symptoms, and the cognitive symptoms within schizophrenia that hopefully you are familiar with. And the role there, of course, you see that the NMDA, again, the glutamate, plays a role. And with a hypofunctional glutamatergic synapse up here, you can end up actually turning down the mesocortical dopamine, turning up turning down the feedback here, and that which ends up turning down the cortical dopamine. So there's an interplay between the glutamatergic circuits and the dopaminergic circuits and the GABAergic circuits that all lead together to having a downstream effect of hyperdopaminergia in the striatum and hypodopaminergia frontally. And that's sort of the evolution, if you will, of thinking about the role of dopamine in psychosis, because we've known since the 1950s, if you block the dopaminergic receptors in the striatum, you can reduce the positive symptoms of schizophrenia. And that led, of course, to the dopamine hypothesis of schizophrenia. But now the argument is really that the dopaminergic effects are the downstream effects of some of the NMDA and glutamate, or the interaction of all of these pathways having an issue. And of course, genetically within schizophrenia, that's why we're also excited by looking at NMDA receptor genes, etc., rather than the dopaminergic, the genes that drive the dopaminergic circuitry directly. Those effects are not as large as the NMDA genetic effects. All right, so this is also a marvelous diagram of these different circuits and what can actually be going on in them, and why the imaging of the subcortical regions can be so complicated in terms of trying to identify what does it mean when we see disconnection or lack of correlation in the bold signal between subcortical regions and cortical regions. So if we look at, for example, sort of starting here from the cortex, much of the input into the striatum is excitatory. From the thalamus back to the cortex is glutamatergic, also excitatory. But once we're down in this thalamus and the striatum and the globus pallidus, etc., and the subthalamic nucleus, then we have this very interesting interaction and interplay of excitatory, inhibitory, disinhibitory. So the overall pathway ends up being disinhibitory, etc. I'm not going to go through this in any too great detail, but there are entire journal issues that are specifically dedicated to the discussion of the EI, or excitatory inhibitory balance, and how that plays a role in brain function, in psychosis in particular. And that is both cortical, but also subcortical, in terms of the interplay between excitatory and inhibitory, and how that leads to these critical balances that we need to have the cognitive processes and the interactions with external stimuli, internal stimuli, timing of thinking, reward prediction, predictive errors, all that good stuff. It all comes down to how all of these circuits really interact, and so much of it is dopaminergic, both driving the excitatory and the inhibitory parts of these circuits. So what does dopamine actually do, right? So the ventral tegmental neurons transmit dopamine in a tonic and phasic mode. The tonic mode is really a consistent, sort of a pacemaker, low frequency activity. The phasic is transient or high frequency modes. And within the striatum, there is very tight, very speedy dopaminergic clearance within the synapse, so that you can have these phasic modes be very tightly controlled. The cortex tends to not be that tight and that speedy. Dopaminergic effects in the cortex can have very long-term effects over the course of minutes, if not longer, whereas the striatal dopamine has very rapid effects. And you can see where, from the computational neuroscience point of view, that people just start itching to say, what is the difference? What does that mean in terms of how these different areas communicate and what the dopamine actually does? Dopaminergic neurons show a number of patterns. We've already talked about the mesolimbic and mesocortical dynamics. And this was, of course, from the Vanderweel paper from 2019, pointing out that functionally distinct populations of dopaminergic neurons within the ventral tegmentum and the substantial nigra continue to be discovered. The story isn't over. The chapters aren't closed. There's increasing evidence for heterogeneity within and between dopamine neuron populations in the processing of rewarding and aversive stimuli, and this calls for more precise interrogations into their cognitive behavioral operations. So there's much more to be able to do, and being able to image these different populations is going to be a key question as we move forward, either from the point of view of being able to use spectroscopy to try to get at the dopaminergic signal, very challenging to do, or use the animal imaging to be able to look at dopaminergic blocking and what effect that has in various animal models, all sorts of questions that we can really ask. Because even if you can image the ventral tegmental area, that doesn't help you because it's not a homogeneous area, and the different subparts of the ventral tegmental area are going to be doing different things. And if your bold signal doesn't have the resolution to get in there, it's going to be tricky to try to infer correctly what the bold signal you're seeing is really drawing from. But a constant model, particularly from the Vanderweer group and others that have published with them, is the idea that what dopamine in the cortex is really doing is improving the signal to noise ratio. And these papers are really this one has had a huge impact and lots of citations. This one more, or these are rather less, so these are sort of older, but you can see over the last 15 years, this idea of dopamine modulating synaptic activity and enhancing signal to noise ratio in the prefrontal cortex, both for aversive stimuli, for appetitive stimuli, or positive stimuli, basically making the prefrontal cortex neurons more efficient, ramping up the signal, ramping down the noise when dopamine is doing what it should do. And a lot of these papers are really computational in nature, so they're simulating the dopaminergic dynamics, simulating the dopaminergic populations, the different inputs, the different outputs, the dynamics of it, and really looking at what comes out of that as you sort of simulate increased dopamine versus decreased dopamine. And then they're also looking at actual human studies and the behavioral impact of these of dopaminergic studies and the aspect of what goes on in these different dopaminergic areas. And the general idea is that dopamine really tunes the prefrontal cortex so that it becomes a more efficient transmitter and allows you to focus. You can see how that leads very quickly to attention and the ability to pull stimuli out of the out of the environment and really identify them, to transmit the information downstream to memory so that you remember things better, etc. One of the more interesting papers that I really like, it's actually from, I want to say 2002. It's actually quite a while ago. Looking at dopamine and cortical efficiency through a very indirect set of methods. So realizing that in the prefrontal cortex, catechol methyltransferase, or COMT, is one of the molecules that is primarily responsible for removing dopamine from the synaptic cleft. So really cleaning up the dopamine, getting it out of the way, getting it back into the neurons so that they can fire again. And making sure that the information is there. That's your COMT. And there's actually a gene and a genotype that affects whether or not this particular form of COMT is more efficient or less efficient. So with different genotypes, some individuals, the dopamine is cleaned up very quickly, and in others, it sticks around for longer. And honest to God, when I'm teaching this in class, I know which one is which, but off the top of my head, I don't remember which COMT genotype is more efficient and which is less. But generally speaking, it does actually seem to have some effects on dopaminergic interactions in the frontal cortex. And in the late 1990s and early 2000s, there was a lot of studies of different COMT genotypes, the individuals with different COMT genotypes, looking at working memory and prefrontal cortex response to working memory, because it does actually seem to have effects in that you can pick up with, relatively large, 50, 100 people, you can actually see genotypic effects with COMT. If looking at by and large, the NBAC working memory task. Now, the NBAC working memory task, for those of you who may have worked with it, can be very demanding or very easy. N, it refers to the number of items that you really have to keep in mind, the number of trials that you have to remember things for. So when N equals 1, it's very easy. When N equals 2, it's a little bit more tricky. When N equals 3, it's really very demanding. And really the limit of N tends to be about 5 or 7. Most people fall apart after about 3 or 4. But if you really work on it, you can get up to 5 or 7. Individuals with schizophrenia have a great deal of trouble with it. They're very impaired with the MBAC task. But they're okay at N equals 1 or 2. At 2 or 3, they really fall apart very rapidly. And it's not uncommon to find in neuroimaging that the dorsolateral prefrontal cortex, basically its activity ramps up the more demand you place in these working memory tasks, in the MBAC tasks. You can see the dorsolateral prefrontal cortex kick in order to maintain performance. So what this study was doing was 2 things. They were looking at COMT genotypes. So they split their people into basically the different COMT genotypes. Are you somebody who's going to be a val or a met? Are you going to be more efficient or less efficient in removing dopamine from your synaptic cleft? And then on top of that, they gave them amphetamine. Isn't that fun? Wouldn't we have all signed up for that one? But they gave people amphetamines, which releases dopamine into, right? It basically forces all of your dopaminergic neurons to kick their dopamine out. So you end up flooding your dopaminergic system. And the folks with very efficient COMT genotypes should be able to clear that a little bit more effectively than the folks who don't clear it out that effectively. But having the dopamine in the system should also mean that you're actually able to do the working memory task just a little bit better. So up to a point, right? Up to a point. And so what they're actually looking at here is the percent signal change in this dorsolateral prefrontal cortex area in different areas. So this is basically where in the brain they're actually looking. The important part is down here, where they can actually show at the one back task. So it's where it's very easy. Basically, on placebo, you have a slight difference between the 2 genotypes. One group's a little bit working a little bit harder than the other one in order to do the one back task at a pretty decent performance rate. So there's a little bit of a difference at baseline. You give them both the amphetamines, and basically their ability to do the task is not that impaired, still capable of doing a one back task, but the bold signal drops off. Really, the frontal cortex is like, you know what? Don't have to work as hard. With the 2 back task, if you look again at on placebo, so sort of at baseline, now you're starting to work harder, and the one genotype is actually having to drive the DLPFC quite hard in order to do the 2 back task, whereas the other genotype is sort of going, the prefrontal cortex doesn't really have to work that hard in order to do the 2 back task. You give them the amphetamines, and now this group that was having to work quite hard doesn't have to work as hard in order to do the task. So far, so good. We're like, hey, amphetamines are great stuff. Now, the problem is now you're doing the 3 back task where you're really having to work quite hard. This group is still able to do the task, but you give them the amphetamines, and suddenly their brain is having a much harder time because they're not able to clear the dopamine as well as they need to. And they've actually gone over the edge. Basically, they've gone over the tipping point of maximal efficiency and now have too much dopamine in their system for doing the task, whereas this group is still showing the sort of positive effect of the amphetamine. So there's a gene by drug interaction here as we get into the more demanding levels of working memory. So again, this is sort of speaking to the interplay not only of the dopaminergic drug, but also and the genotypes, and the circuitry to say what is it that's really going on with the dopamine. But the argument is it up to a point, it makes the prefrontal cortex more efficient. Depending on who you are and where your dopaminergic levels are, you can basically end up going over the edge, having too much dopamine over that U-shaped curve. You end up with too much dopamine and or too little, and either way, you're not performing at maximal efficiency in your prefrontal cortex. All right. So in conclusion, basically, when we're talking about the role of dopamine, we want to think about it from the point of view, certainly in the cortex, of really tuning the neurons, thinking about it from the point of view of increasing the efficiency of the network. But you don't, if you have too much or you have too little, you end up sort of not being at that maximal efficiency. But you do have to consider the complexity of the dopaminergic action by which neuron you're dealing with, which receptor you're dealing with, which brain region you're dealing with, which network, what is the overall function of the network, overall, what are the dynamics of the network. And the recommendations within the papers that we were looking at is we want to consider which networks are dysfunctional really by patient rather than by diagnosis. So rather than saying, oh, within schizophrenia, it's this network or it's that network, within patients, you might see differences based on their genotype and their wiring as to which of these networks are really dysfunctional. So they were considering that you might want to split your patients with schizophrenia into those who have striatal hyperdopaminergia versus those for whom the striatal hyperdopaminergia is really not the key problem, but it might be the other networks that are the issue. And also to consider the substantia nigra as a therapeutic target to say, can we actually get to the sort of the core of the problem by looking at the substantia nigra, taking into account, of course, its distinctions. And that's pretty much everything I wanted to say. So I'm going to stop sharing and hope that we can have some questions from there. Marvelous. So first of all, are any questions or feedback on that?
Yeah, I have one. Thank you for that. That was really really useful to think about these circuits and very comprehensive. I'm curious about, we kind of have talked about this neural variability and kind of this sort of came out of that. And it seems like a lot of what you're talking about is sort of the inhibition excitation balance, and there's sort of being an optimal level before you sort of go over the edge, as you put it. I'm curious what you think the link might be with some of that.
I have a sort of gut feeling about it. I know that there are some papers that have actually dealt with that sort of looked at the dopaminergic effects on the variants. And when they're talking about the amplifying the signal to noise, largely what they're talking about with the dopamine is this, I'm trying to remember if it's the tonic or the phasic effect, of reducing the background variability. So reducing the background variability so that the responsiveness to the signal can be less variable, so that the phasic response, in a sense, they're sort of increasing the tonic background to reduce the variability and then increasing the sensitivity to the phasic response. And what that means, I think, in the sense of variability, is that it probably, we were talking before about how having some underlying variability is a good thing, right? But there's such a thing as too much, right? And so you don't want so much variability that any responsiveness to the stimuli gets wiped out, but you don't want so little that every response to the stimulus is identical, right? You wanted some variability. And I think that's where this dopaminergic balance is really going to play a role.
Right. Yeah. And I'm curious too about sort of, we talked about regional effects as well. Do you
Hmm.
think this could account for some of those differences too, in terms of sort of more perhaps lower level sensory responses differing from more higher level
Absolutely.
cortical ones and so on?
Absolutely. Yeah, absolutely. Especially when you consider all of the links and all of the synapses in those circuits, right? And the differences between what's going on in the sort of lower level subcortical areas. And so genetic differences, environmental differences can really lead to different processing there that can have almost independent effects of what's going on downstream in the cortex. So you could see individuals with striatal balances of dopamine excitation, inhibition, et cetera, that are going to have one pattern and the cortex can do something else and vice versa. So yeah, I think you would see some very regional differences that way, just because there are different dopaminergic pathways that have different dynamics.
Right. I have a few other questions, but I'll sort of wait.
Yeah, yeah.
I have just a question. Can we talk a little bit more about what's causing this inhibition or excitation of dopamine? How are we getting here?
Oh, let me start. So you saw this. Let me share the slides here. You saw this thing. Let's see if I can go back. It comes, well, let's not do that one. Let's go to this one,
You're
right?
right. So how are we applying the brake and how are we applying the accelerator in terms
Okay,
of like what happens before that?
right. Well, there's a certain amount of miracles up here, right? In the sense that we're not really, if you think about the brake, you're basically saying, well, the glutamate here, the glutamatergic projections to the midbrain, to the GABA interneurons in the midbrain are overactive. Why are they overactive? Well, that sort of pushes the question back a bit. But if they are overactive, they are basically overdriving the inhibition of GABA, which then is inhibiting the dopaminergic output so that specifically to the mesocortical projections, to the dopaminergic neurons that are part of the mesocortical projection. So you end up with too little dopamine in the cortex, right? On the other hand, the accelerators, the glutamatergic projections from the cortex to the midbrain that are actually projecting to the dopaminergic neurons, which are going into the frontostriatal, mesolimbic, and nigrostriatal circuitry. And in that case, there's too much dopamine. So you can have, because you have these multiple loops within the distinctions within the ventral tegmentum and multiple loops from the cortex, you can actually have two independent effects. Now, why is it out of whack, right? How does it get out of whack? Well, that's where you have a much bigger kind of question in terms of is it a genetic issue? Is it a combination of genetics and environment? And what is it that's actually regulating this so that these receptors aren't quite working correctly? And the folks who had this hypothesis actually were basically saying, look, the key issue, their hypothesis is that the key issue is in the NMDA receptor system. That the NMDA receptor system on the glutamate neurons is slightly faulty, not completely faulty, but slightly faulty. And so what that does is it actually fails to turn off the projections to the midbrain, so that this faulty glutamatergic synapse basically fails to turn this off, so that then this is overactive, and then this overdrives the brake, which is what overdrives the mesocortical thing. So they're basically going back to the NMDA and saying, because it's not a dopaminergic problem, really it's an NMDA problem. And so the signaling within the cortex sets up a loop where you end up with too little dopamine just because of the cortex firing. So that's one hypothesis, right? Then that's being looked at quite closely.
Okay, but that's just a hypothesis of why glutamate is having this role. We're
Exactly.
not concrete on it. Okay, great.
Well,
Thank you.
in the sense that, I mean, there's evidence, right? I mean, there's lots of evidence for the glutamate hypothesis regarding these dysfunctions. There's animal studies, there's genetic studies, there's circuitry studies. Um, what is that? But does that explain everything? You know, nothing explains everything.
Right.
It probably is playing a role in some of the clinical presentations of schizophrenia. And then you ask, well, why don't we have a glutamatergic target for schizophrenia? And the problem is glutamate is unlike dopamine, which is in very specific parts of the brain, right? Dopamine is largely here. Glutamate's kind of everywhere. So you can block dopamine, and most of your action will happen, here and to a lesser extent here, particularly if you're blocking, D2 receptors in here, you'll have less of an effect everywhere else. You block glutamate, people just keel over, right? It just doesn't work well. Everything stops. So you have to figure out, how do you get the glutamate to the particular synapses that you really want to modify and modulate. And believe me, there are people trying to figure that out.
Great, thank you.
Okay, so Amin asks, how many studies have been done looking at the 3 loops in schizophrenia using functional MRI? Directly or indirectly? Because indirectly, of course, you can say all of them, right? Even the big ICA connectivity studies where we've looked at just 100 components, all of Eshwar's papers looking at dynamic and static functional connectivity, where you identify that you've got a subcortical module and cortical modules, and can sort of support these kinds of findings. It says, the subcortex is not as connected to the cortex as it should be. The cortex is not connected to the cortex as it should be. But getting into, are we looking at mediodorsal versus lateral thalamus? I don't know that we certainly don't do that. And ICA tends not to identify those as different components, but there are certainly studies that use seeds, that specifically put seeds in very specific areas of the thalamus to see what the differences are. And doing that, you can certainly start to tease some of these networks apart. It helps to have the newer, higher resolution resting state data, but even with some of the older resting state data, if you seed it specifically from different parts of the thalamus, because the thalamus is this big chunky area, or as you were seeing from the paper that I pointed out about the caudate, looking at very specific parts of the dorsal striatum, you can certainly do it. There's lots of studies there. Yeah, go ahead.
Thank you. The reason I'm asking is because we can directly model these. So we can
exactly
do a modeling, and we don't even need to have like a very high, it's great if you can have like a subregion of the thalamus and caudate, but even if we pick the thalamus and model them at the same time, we can see how these connection and loops change between, for example, healthy and, this typical control schizophrenia.
Yeah.
That's why I was asking.
Definitely.
I was wondering how interesting can it be from your view?
Oh, I think it's a critical question, and certainly as I was sort of skimming the literature trying to figure out which articles to suggest for you guys, there were lots that were doing large-scale simulations of dopaminergic networks and dopaminergic pathways, and trying to say what is it that actually happens when stimuli come in, and how do we actually quantify what the differences are between functional D1 receptors versus functional D2 receptors, et cetera, and what would that mean in terms of the circuitry and the cortical pathway. And from those, we could probably infer functional MRI results, because certainly some of them were at that point. So yeah, that's an active area of research, I think, and there's a lot of things that could be done there. Definitely happy to chat with anybody who wants to take that on.
Hello, Dr. Turner. Thank you so much for this excellent presentation. Really enjoyed it. I just had a couple of quick questions or discussion topics to broach.
Sure.
As you know, I did cognitive testing for many years there at UC Irvine, and so I tested hundreds of schizophrenia patients for our clinical trials and for the Burn study. And one test that always struck me as incredibly sensitive to the impairments we see in schizophrenia was the Wisconsin card sort test, and that's well known, as a measure of executive dysfunction. And it always made me wonder, because I saw many times the patients, they would find the right set. And for those of you who don't know, you have 4 cards at the top of the page, each with a different shape, a different color, and a different number of symbols. And then you give the patient 1 card at the bottom, and they have to choose one of the 4 that it matches. And you don't tell them how it matches, but it can either match by color, shape, or number. And so patients, a lot of times, they'll get the first set. They have to, play around for a while until they find one that matches. And generally, you'll see them, achieve at least 1 set. And then after they've done that for 10, then the test switches. So you have this set switch. And so what you see in patients with schizophrenia is a lot of times they'll ignore, or even they'll say to me, I don't know why it's saying I'm wrong. I'm getting it right. So it's like they're not attending to the negative reinforcer, and they persist or perseverate with the set that they've already achieved. So it's like the reward is more important than the negative information they're receiving. So I'm wondering if anyone has used the Wisconsin card sort. And I know people have seen the association with schizophrenia in many different ways, but I didn't know if anyone had used that as an activation paradigm.
There have been a lot of fMRI studies of the Wisconsin card sort. It fell out of disfavor because it was so cognitively complicated. Right? I mean, even as you describe it, you have to generate a hypothesis, you have to test the hypothesis, you have to integrate the feedback, and then make a decision as to what to do next. And that's just too many cognitive processes for the purposes of interpreting what's actually going on. And so I think it's been, for the fMRI, you'll find in the literature over the last decade, there have been people addressing, specifically the set switching aspect, or specifically the hypothesis generating aspect, or specifically the integration of negative feedback aspect, or things like that. So to try to tease apart which of these circuits is really involved with which. But certainly the inability to integrate the negative feedback is got to be dopamine. I mean, certainly the Vanderweel team would argue that's dopaminergically moderated in the prefrontal cortex. Your ability to really focus on that was wrong, do something else, is a dopaminergic issue. Now, but there's also the issue that generating new hypotheses is also, the creativity aspect of trying to say, what can I try next? And motivating yourself to do that is also not a simple process.
Right. I have one other quick question about, it's been a year since I was in the drug development, field. But at the time, everyone was talking about the metabotropic glutamate receptor 2,3 agonist. I think Lilly had an mGlu 2,3 agonist. I just wonder if if they're still in testing with that. And
my understanding is that's one of those glutamatergic drugs that looked promising and then fell apart. Yeah, because it works great in animals. It does exactly what it's supposed to do, and it's the targeting of the glutamatergic neurons. I think it really highlighted, if I recall correctly, it or one of the other glutamatergic drugs basically highlighted, if you give it to somebody, their whole system falls apart. You get all the side effects were just so bad. That it wasn't worth, the cure was worse than the disease. So, yeah, I think I don't think people have given up on it, but certainly, how long ago was it that so many of the major pharmaceutical companies basically said, we're stepping out of CNS drugs, we're not going to be developing antipsychotic drugs. It's too hard. The systems are too complicated. And that's where the neuromodulation really came to the fore. People are like, well, what can we do with TDCS and TMS that can be a little bit more targeted in some cases? To say, we can't chemically hit the synapses, but maybe electrically we can hit the synapses and do something that way. It's anybody's game.
As you know, we did the study with Jen. We were targeting the DLPFC in schizophrenia with our TMS. And I do still have I did the neuropsych testing before and after those procedures, and I still haven't fully dug into that data. I've got it in a folder here somewhere. Yeah, but I'm pretty sure we did. No, I don't think we had the Wisconsin card sort on that. Otherwise, it would be really interesting to go back and look at.
Yeah, exactly. But memory tests are all the same.
We did that on the burn study, so that's something that could be mined from that data set as well, looking at relationship to different scores on the Wisconsin card sort.
Exactly.
Okay, well, thank you. Very thought-provoking. I appreciate it.
Oh, glad you could join in. Aaron, for those of you who haven't met Aaron, and of course most of you haven't, he and I worked together on the F-Bird many, many years ago back in California, and he's now a graduate student in bioinformatics at the University of Arkansas in their bioinformatics program. Right? That's where
Yes, that is
that's
correct.
it. Yes. So, so those of you who are interested in bioinformatics or computational approaches or information management or large scale analyses, Arkansas is an interesting place to check out. So,
Indeed. Thank you.
all righty, thanks. Jeff, are we good?
Well, yeah, we're at 10:55. I do have more questions, but I also don't want to take any more of everyone's time. So I did just want to thank everyone for joining today. I know it's really late in the semester, so I appreciate that. And Dr. Turner, thank you so much for giving this really informative session.
I hope it was helpful.
Yeah, I think it really fit nicely with the conversations we've been having about ability and functional connectivity. And there was actually a suggestion about introducing fMRS in the session, which I think, again, sort of thinking about these different levels of these neurotransmitters, it might be useful to talk about that in the future.
Oh, definitely. And again, that's one of those more cutting edge techniques, because MRS for so long was just okay, scan them for 20 minutes, just to get the estimate of what the transmitter was, over the last 6 weeks. It's really an integrated, effect. And the idea that you can actually look at functional spectroscopy over the course of, seconds or minutes is just astounding.
Yeah. Is that an active area in this? I imagine it must be.
Oh, very much so. And actually, Jenny Robinson at Auburn is one of the key people in it. So I think she'd be happy to give a presentation or have some of her students give a presentation. One of her PhD students who just graduated gave a presentation last fall in my lab on functional spectroscopy and sort of looking at how things change over time. Over minutes in a scanner, just in their 7T. It's really impressive. I hope Kelly and Tom, do you remember that? Wasn't that what he was talking about?
I honestly don't remember. I'm sorry.
Well, I recorded it for just that reason. We have it. We can actually play that again at some point. But yeah.
That would be great. Yeah, and I think just kind of as an announcement as well, there are a lot of open spots for the spring. So please do get in touch if you have a topic you'd like to talk about or a paper that's come out recently that you think would be good to discuss. Just as a reminder, we'll have our cluster correction workshop at the end of January with Dr. Andy Young. But February is open, as are many other months in the spring. So
wait, now are we going to move the time at all?
So right now we have Andy's workshop for 10 a.m. on Tuesday, the 26th. But definitely if sort of generally for next semester, if other times work better with people's schedules, we can.
Well, my class is Tuesday morning.
Okay.
So I'm actually looking at the syllabus and trying to figure out how I can get to cluster correction by the 26th, which I can't. But I will probably boot people out of class to go attend the workshop no matter what. But I can't make Tuesdays at 10 regularly.
Okay, yeah, we can. I'm definitely open to other times. We could maybe put another survey on
a
Slack.
doodle poll or something.
Yeah.
So see what we can do. Perfect. Yeah, because I know several of my students are in that class as well, so they wouldn't be available Tuesdays at 10 either.
Okay.
Let's find a time. Marvelous. And Aaron, obviously, spread the word if other people in your program want to join in. They're welcome to join into the brain space talk.
Yes, I know Dr. Larson Pryor really wanted to join today, but she had a prior commitment. So she's reminding me to go and take notes.
Excellent. All right. Well, and I'm happy to send you my slides. So yeah, if she wants me to email the slides, I'm happy to do that.
Okay. Yeah, that would be helpful. Thank you.
Great. All right. Well, let's call it a day then, right?
Yeah. Thank you, everyone.
Thanks all. Have a good
Happy holidays, everyone.
Bye now.